Good morning students. We will continue the carbohydrate metabolism and today's topic is HMP Shunt Pathway. Long form of HMP Shunt Pathway is hexose monophosphate shunt pathway. Why it is called as shunt? Because normal pathway of glucose utilization is glycolysis. But after meal, when the blood glucose level or the glucose inside the cell is high, at that time some glucose is diverted in this pathway. Therefore, it is called as shunt pathway. The synonyms of these pathways are pentose phosphate pathway, Dickens-Horker pathway, shunt pathway, or phosphogluconate oxidative pathway. So these are the various synonyms of this pathway. This pathway occurs in two phases, oxidative phase and non-oxidative phase. Non-oxidative phase is the continuation of oxidative phase. Site. Oxidative phase of this pathway occurs in the cytoplasm of liver, adipose tissues, adrenal cortex, mammary gland, testes, ovaries, RBCs, lens of eye, etc. Whereas non-oxidative phase of this pathway occurs in all tissues of the body. So this is the complete HMP Shunt pathway. It starts from glucose to the ribulose 5-phosphate. That part of pathway is called as oxidative phase of this pathway, and beyond this ribulose 5-phosphate, it is called as non-oxidative phase of this pathway. In non-oxidative phase of the pathway, generally interconversions of various sugars are seen. One more point about this pathway is that, especially about non-oxidative phase of this pathway, is you cannot study single pathway, because during non-oxidative phase many intermediates of one pathway get combines with intermediates of other HMP Shunt pathway and they form new sugar. Therefore, you cannot study single HMP Shunt pathway like glycolysis. If you take one molecule of glucose, that will give two molecules of pyruvate. In this way, you cannot study this HMP Shunt pathway and especially the non-oxidative phase of this pathway. So we start the pathway. Glucose 6-phosphate undergoes oxidation to form 6-phosphogluconolactone. The enzyme required for this step is glucose 6-phosphate dehydrogenase. Here two hydrogen atoms from this glucose 6-phosphate are removed and they are accepted by NADP to form NADPH. Up till now we have studied hydrogen acceptors are NAD or FAD. Here the hydrogen acceptor is NADP, which is a coenzyme at this step, and that NADP is reduced to NADPH. Now this glucose 6-phosphate dehydrogenase is the rate limiting enzyme of this pathway, or it is a important enzyme or allosteric enzyme of this pathway. Next, 6-phosphogluconolactone with the help of enzyme hydrolase get converted into 6-phosphogluconate. This 6-phosphogluconate undergoes second oxidation. Again, two hydrogen atoms are removed. At this step, one molecule of carbon dioxide is also removed. So it is actually oxidative decarboxylation occurs at this step. And the enzyme required for this step is 6-phosphogluconate dehydrogenase. Again, the two hydrogen atoms which are removed from the substrate are accepted by NADP coenzyme and get reduced to NADPH, and the product we get is ribulose 5-phosphate. So up to this step, the pathway is called as oxidative phase of the pathway. Now the non-oxidative phase of the pathway starts. Now the ribulose 5-phosphate which is formed either get converted into xylulose 5-phosphate by enzyme epimerase or get converted into ribose 5-phosphate with the help of enzyme isomerase. This conversion takes place according to the need of the cell and the concentration of xylulose 5-phosphate and ribose 5-phosphate in the cell. Now, xylulose 5-phosphate which is formed in one pathway combines with the ribose 5-phosphate formed in another pathway. I have already told you that you cannot study single HMP shunt pathway. So here the product of two pathways, xylulose 1,5-phosphate and ribose 5-phosphate. Now these two sugars with 5 carbon get condensed and gives pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. The enzyme required for this step is very important, called as transketolase. Now this transketolase condense this ketose sugar with 5 carbon and aldose sugar with 5 carbon, that is xylulose 5-phosphate and ribose 5-phosphate, to form 7 carbon pseudoheptulose 7-phosphate and 3 carbon glyceraldehyde 3-phosphate. Now this transketolase enzyme requires a coenzyme thymine which is vitamin B1. If a patient is suffering from vitamin B1 deficiency, then the activity of this enzyme transketolase is also decreased. So this is the clinical importance. Now in the next step, pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate, 7 plus 3 carbon, get condensed to form erythrose 4-phosphate and fructose 6-phosphate. Means 4 plus 6 sugar is formed. 7 plus 3 sugar condenses and form 4 plus 6 sugar with the help of enzyme transaldolase. Now fructose 6-phosphate which is formed get converted into glucose 6-phosphate, and the erythrose 4-phosphate which is formed on the left side get combines with the xylulose 5-phosphate of the another pathway. Here the third pathway is involved. Now 4 plus 5 carbon sugars condense to form fructose 6-phosphate and glyceraldehyde 3-phosphate, means 6 plus 3 sugars are formed. Again the fructose 6-phosphate get converted into glucose 6-phosphate, and glyceraldehyde 3-phosphate which remains here combines with the glyceraldehyde 3-phosphate of another pathway. That means here again fourth pathway is involved to form again fructose 6-phosphate, glyceraldehyde 3-phosphate, 2 molecules, means 3 plus 3 sugar combines to form 6 carbon fructose 6-phosphate which get ultimately converted into glucose 6-phosphate. So this is little bit difficult to remember. This is the trick of non-oxidative phase. 5 plus 5 sugar, 5 plus 5 means 5 carbon plus 5 carbon sugar combines, that means xylulose 5-phosphate and ribose 5-phosphate combines to form 7 plus 3 sugars, that means pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. Now this 7 plus 3 sugar condenses to form 6 plus 4 sugar, that is fructose 6-phosphate and erythrose 4-phosphate. Now this fructose 6-phosphate get converted into glucose 6-phosphate. Now the 4 carbon erythrose 4 phosphate then combines with 5 carbon cellulose 5 phosphate of other pathway. So here 4 plus 5 sugar combines to form 6 plus 3 fructose 6 phosphate and glyceraldehyde 3 phosphate sugars. That fructose 6 phosphate is again converted into glucose 6 phosphate. Now the glyceraldehyde 3 phosphate of this pathway and glyceraldehyde 3 phosphate of another pathway, that means 3 plus 3 condense to form fructose 6 phosphate again, which is converted to glucose 6 phosphate. So in this way you can remember 5 plus 5 gives 7 plus 3, 7 plus 3 combines and gives 6 plus 4 sugar, 4 plus 5 sugar combines to form 6 plus 3 sugar, and this 3 plus 3 sugar combines to form 6 carbon sugar. In the book the pathway is given like this. They have given, it is not a single pathway, they have taken 6 molecules of glucose 6 phosphate at a time, which get converted to 6 molecules of phosphogluconolactone. 6 phosphogluconolactone get converted to 6 phosphogluconate. After oxidative decarboxylation, that 6 molecules of phosphogluconate get converted into 6 molecules of ribulose 5 phosphate. Now here the interconversion starts. Now out of 6 molecules of ribulose 5 phosphate, 4 molecules epimerize to form cellulose 5 phosphate and 2 molecules isomerize to form ribose 5 phosphate. Now these 2 molecules of ribose 5 phosphate then combines with the 2 molecules of cellulose 5 phosphate. Out of 4, 2 molecules of cellulose 5 phosphate combines with 2 molecules of ribose 5 phosphate to form glyceraldehyde 3 phosphate 2 molecules and 2 molecules of pseudoheptulose 7 phosphate. So 5 plus 5 sugar, cellulose 5 phosphate and ribose 5 phosphate, 2 molecules of each condenses to form 2 molecules of glyceraldehyde 3 phosphate and 2 molecules of pseudoheptulose 7 phosphate. Now these 7 plus 3 sugar condenses to give 2 molecules of erythrose 4 phosphate and 2 molecules of fructose 6 phosphate. Now this 2 molecules of erythrose 4 phosphate then combines with the 2 molecules of cellulose 5 phosphate which are remaining there. Out of 4 we have utilized only 2 to combine with ribose. So 2 cellulose 5 phosphate are remaining. Now these 2 cellulose 5 phosphate combines with 2 molecules of erythrose 4 phosphate, that is 5 plus 4 sugar, condenses to form 2 molecules of fructose 6 phosphate and 2 molecules of glyceraldehyde 3 phosphate. These 2 molecules of glyceraldehyde 3 phosphate get condensed to form 1 molecule of fructose 6 phosphate. So in this way 5 molecules of fructose 6 phosphate are regenerated at the end of the pathway, and we have taken 6 molecules of glucose 6 phosphate as a substrate. Here there is a net loss of 6 carbon, and these 6 carbon atoms are lost in the form of carbon dioxide in the third step, which is called as oxidative decarboxylation step, where the enzyme is 6-phosphogluconate dehydrogenase. Therefore 1 fructose 6 phosphate is less. In the exam you can write down this pathway or you can write down this pathway. Now come to energetic part of this pathway. Here no ATP is synthesized, no ATP is utilized, because in our body reduced NADPH does not give any ATPs. NADH will give 2.5 ATPs, but reduced NADPH will not give any ATP in human beings. Next, regulation of pathway. As I have told you, glucose 6 phosphate dehydrogenase is the rate limiting enzyme. So the reaction catalyzed by this enzyme is called as rate limiting step of this pathway or HMP Shunt pathway. That means If you stimulate this enzyme, the whole pathway is stimulated. If you inhibit this enzyme, whole pathway is inhibited. First, about hormonal regulation. As I have already told you, this pathway occurs when blood glucose level is high, when the cellular glucose level is high, which is generally seen after meal, and the hormone which is present in the post meal state is insulin. So insulin stimulates this pathway, whereas adrenaline and glucagon inhibit this pathway. After hormonal regulation, come to the allosteric regulation. The stimulator of the enzyme G6PD is NADP. When the NADP level is more, then they stimulate the glucose 6-phosphate dehydrogenase pathway, so that NADP is utilized and get converted into NADPH. At the same time, NADPH level when increases, it acts as a inhibitor of the enzyme glucose 6-phosphate dehydrogenase. At the same time, when the concentration of pentose sugars inside the cell increases, they also act as a allosteric inhibitor of this glucose 6-phosphate dehydrogenase enzyme. Significance of this pathway. This pathway gives us two important components. First is NADPH and second is pentose sugars. So we will see the importance of these two. First, importance of NADPH for human body. First important thing is NADPH is required for synthesis of cholesterol, fatty acid, steroids, etc. NADPH also required for scavenging the free radicals from the body. It is also required to maintain the RBC membrane integrity. NADPH prevents the formation of methemoglobin, or it prevents the accumulation of methemoglobin. NADPH plays an important role in detoxification of drugs. It preserves lens transparency in eye. It has some bactericidal activity in macrophage. So we will study in little detail about the various actions of NADPH. First, about free radical scavenging. Free radicals are, examples I have given: superoxide, anion, hydrogen peroxide. These are free radicals. Free radicals means any compound with unpaired electron is called as free radical. So these free radicals are constantly formed in the body, and they have to constantly removed out from the body. If they get accumulated, then they causes destruction of DNA, protein, fatty acids and all biomolecules. Therefore, they may cause cell death. In our body, to remove the free radicals, the enzyme system is there, and this enzyme system is constituted by three enzymes. First is superoxide dismutase, peroxidase and glutathione reductase. We will see the actions of this enzyme in the next slide. So this is the slide showing actions of these three enzymes. Now first see O2 dot. Dot indicates that oxygen is a free radical. If you are writing a dot, superscript dot on any compound, then superscript dot indicate that compound is a free radical. That means it has unpaired electron. So two superoxide anions are there. The enzyme SOD, superoxide dismutase, convert this superoxide anion into hydrogen peroxide and oxygen with the help of two protons. Now this hydrogen peroxide is also damaging for us. Therefore, the another enzyme POD, that is called as peroxidase, acts on this H2O2 and convert it into water. To convert H2O2 into water, this peroxidase enzyme or POD requires two hydrogen atoms, and these two hydrogen atoms are supplied by reduced glutathione. GSH indicates the reduced form of glutathione. So two molecules of glutathione, reduced glutathione, supplies two hydrogen atoms in the reaction, and the POD will convert this hydrogen peroxide into water. But during this process, the reduced glutathione, that is GSH, two molecules of GSH, get converted into GS. That GS is the oxidized form of glutathione. GSH indicates reduced form of glutathione, whereas GS indicates oxidized form of glutathione. Therefore, this glutathione should be reduced back so that it can again supply two hydrogen atom for peroxide and the cycle continue. To convert the oxidized form of glutathione into reduced form, the enzyme required is glutathione reductase. This glutathione reductase takes two hydrogen atom from NADPH and add it to the glutathione, that is oxidized glutathione, and convert it into reduced form. So NADPH here plays a role by donating the hydrogen atom. So this is the importance of NADPH, and NADPH we are getting in HMP Shunt pathway. So in this way, the NADPH helps to remove the free radicals from our body. NADPH also helps to maintain the RBC membrane integrity, simply because in the RBCs the concentration of hydrogen peroxide is more. That means formation of hydrogen peroxide is more as compared to other cells. If this hydrogen peroxide get accumulated, it may damage the cell membrane and leads to breakdown of RBC, that is hemolysis. But NADPH along with this enzyme, glutathione reduced glutathione and this enzyme helps in removing the hydrogen peroxide. The mechanism is same. The hydrogen peroxide is removed by the same mechanism, same enzyme. So NADPH helps to maintain the erythrocyte membrane integrity. NADPH prevents methemoglobin accumulation. What is methemoglobin? Methemoglobin means a hemoglobin containing iron in ferric form. Normal hemoglobin contains iron in ferrous form, which is a hexavalent. But when that ferrous iron is converted into ferric iron by the free radicals present in our body, that ferric iron is pentavalent, it cannot carry oxygen. Therefore, this ferric iron should be converted into ferrous form again, and they require an electron to convert the ferric into ferrous. Electron is required, and that electron is supplied by reduced NADPH, so that ferric form is converted into ferrous form. Remember, iron with ferrous form is called as hemoglobin, and iron with ferric form is called hemoglobin with ferric form is called as methemoglobin. So conversion of hemoglobin to methemoglobin is continuously taking place in our body by free radicals, but the same is converted back to normal hemoglobin with the help of electrons donated by NADPH. So NADPH prevents accumulation of methemoglobin in the body. NADPH also plays an important role in detoxification of drugs. Detoxification of drugs means conversion of insoluble form of drug into soluble form so that they can be easily excreted from the body. That is called as detoxification. Now, majority of the drug we are taking, they are hydrophobic in nature. They are made hydrophobic so that they can enter in any cell directly without any transporter, because cell membrane will allow the hydrophobic compounds easily as compared to hydrophilic compounds. Therefore, the drugs are made hydrophobic. But once the action of this drug is over, they have to be excreted from the body. Otherwise, they get accumulated inside the cell. To remove these drugs from the body, we have to convert this hydrophobic form of drug into hydrophilic, because body will only excrete hydrophilic substances. Now, an enzyme system is there which consists of many enzymes. That enzyme system is called as liver microsomal 450 enzyme system. So these enzymes present in the liver convert the hydrophobic form of drug into hydrophilic form. I have shown here, and majority of them will add hydroxyl group, OH group to the drug, and due to addition of this hydroxyl group to the hydrophobic compound, it becomes amphipathic and it gets soluble in the water and can be easily excreted. Now, to convert this hydrophobic form into hydrophilic form of drug, the enzyme requires hydrogen atoms which are supplied by NADPH. So in this way, NADPH helps in detoxification of drugs. Again, the concentration of NADPH is maximum in the lens of eye, and this is also important because the free radicals which are formed inside the lens, if they accumulated, they causes damage to the lens and the lens becomes opaque, or the transparency of the lens is lost. To maintain the transparency of the lens, the free radicals should be removed early, and NADPH, we have seen, with the help of the three enzymes, removes the free radicals from the lens. Now the next function of NADPH is in the macrophages. It helps the macrophage to kill the bacteria. Now, it is observed that macrophages and PMN, polymorphonuclear cells, that means mainly neutrophils, when these cells engulf a bacteria, their target is to kill the bacteria. So when these cells engulf the bacteria, they respire more oxygen. That is called as respiratory burst. They will take more and more oxygen, or they will increase the concentration of oxygen inside the cells. Now, this oxygen is then converted into free radical, that is superoxide anion, by adding a electron donated by NADPH. So enzyme at this step is called as NADPH oxidase, which convert the oxygen into superoxide anion by adding a unpaired electron from NADPH. Now this superoxide anion is acted upon by the enzyme superoxide dismutase, and it will convert it into H2O2 that we have seen in the previous slide. Now this hydrogen peroxide combines with chlorine with the help of enzyme myeloperoxidase and form a compound HClO, which is called as hypochlorous, and this HClO then kill the bacteria. So if you see on the left side, NADPH obtained from HMP Shunt pathway act as a donor of unpaired electron to the oxygen to form the superoxide anion. So here the role of NADPH is not in removing the free radical, but the role is to generate the free radical to kill the bacteria. So these are the various important role of NADPH in the body. Now we will see the importance of ribose sugar or pentose sugars which are formed in the HMP Shunt pathway. All of you know ribose and deoxyribose sugars are required for synthesis of RNA and DNA. At the same time, these sugars are also required for the synthesis of various nucleotides like ATP, GTP, CTP and some coenzymes. So this is the importance of ribose sugars. Now come to the clinical aspects of HMP Shunt pathway. The first and most important disease related to this pathway which is seen in clinical practice is G6PD deficiency or GPD deficiency, glucose 6-phosphate dehydrogenase deficiency, which will lead to hemolysis. How it leads to hemolysis we will see later on. First, about this disease. It is the most common disease seen in clinical practice. It is transmitted by X-linked recessive trait. If G6PD deficiency is there or glucose 6-phosphate dehydrogenase is deficient in any person, then the rate of HMP Shunt pathway in that person is lower. Therefore, generation of NADPH in this person is found in low quantity. As the NADPH is required for removal of free radicals and the availability of NADPH is low, the free radicals are not readily removed from the cells, and these free radicals will damage various cells. The most vulnerable cells which can be easily damaged by free radicals is RBC. So the patient with NADPH deficiency generally present with hemolysis. And the typical history of this patient is the patient may have taken some antimalarial drugs like Primaquine, and after taking that drug Primaquine, after 3 to 4 days he develops anemia, jaundice due to breakdown of RBCs or hemolysis. Why this is? Because Primaquine and other oxidative drugs, when they are taken, they stimulates hydrogen peroxide formation. They increases the H2O2 formation in the body. When the hydrogen peroxide concentration in the cell increases, especially in the RBCs, and there is low NADPH level, that is low free radical scavenging activity in such person, then such RBCs are easily undergoes hemolysis, and the patient present with anemia and jaundice in patients with glucose 6-phosphate dehydrogenase deficiency. Not only Primaquine, some toxic glycosides or fawabins, drugs like sulfa drugs, which is antibiotic, furadentine, it is also antibiotic. So these drugs also leads to hemolysis. The mechanism is same. All these drugs are oxidative drugs. They increases the formation of hydrogen peroxide inside the RBCs. Due to deficiency of enzyme G6PD, the NADPH level in the RBC is very low. Therefore, RBC cannot remove the excess of hydrogen peroxide which is formed due to this drug, and this hydrogen peroxide get accumulated and damage the red blood cell, leading to hemolysis. After hemolysis, the patient will have jaundice because hemoglobin is converted into bilirubin. So bilirubin concentration increases, and as the concentration of hemoglobin decreases, the patient is having anemia. geographical distribution of G6PD deficiency correlates with malarial endemicity. Endemicity means where the cases of malaria are more in that area, then that area is called as endemic. In the study it is found that where the malarial cases are more or malarial cases are endemic, in that area the chances of finding G6PD deficiency persons are more, or G6PD deficiency in people are seen in area where malarial cases are more. The deficiency of enzyme G6PD in the red blood cells leads to resistance to the plasmodium infection, because plasmodium infection requires reduced glutathione for their life cycle, and plasmodium will grow in two tissues, first in liver and in RBCs. So in RBCs, when they complete their life cycle, they require reduced glutathione to complete their life cycle. But in G6PD deficiency persons, the concentration of reduced glutathione is low, and hence the malarial parasite will not survive in this person, and this person will not get malaria, or you can say these persons are resistant to plasmodium infection. So in this way G6PD deficiency saves the people from malaria. Next clinical disorder is methemoglobinemia, that is accumulation of methemoglobin. We have already studied hemoglobin is converted into methemoglobin by free radicals simply by converting the ferrous iron into ferric form, and that ferric form is again converted back to ferrous form with the help of NADPH. So in G6PD deficiency the NADPH concentration is low, so reconversion of methemoglobin to hemoglobin is at lower rate as compared to its formation. So backward reaction is at lower rate as compared to forward reaction. So methemoglobin get accumulated and the person will have methemoglobinemia. Transketolase deficiency, it is also one of the enzyme of HMP Shunt pathway. I have already told you this enzyme requires coenzyme thymine for its activity. If person is suffering from transketolase deficiency, then if such person is exposed to alcohol or exposed to thymine deficiency, then that person will develop a disease called as Wernicke-Korsakoff syndrome or encephalopathy. It is a brain disease. If we want to measure a thymine deficiency of a person in a person, then you cannot measure a thymine level in the blood, but we have to study the transketolase reaction, or we have to study the transketolase activity of that person. By knowing the transketolase activity in the person, you come to know about the thymine status of that person. Sometimes a case is asked on this topic, like this 34-year old man was treated with primaquine. 4 days after he developed anemia, hematuria and decreased hemoglobin level from 11 gram deciliter to 7 gram deciliter with appearance of jaundice. Whenever there is a case like this where you see hemolysis plus any drug like primaquine, then it is a definite case of G6PD deficiency. So the questions on this case are: which enzyme is deficient? Enzyme deficiency is glucose 6-phosphate dehydrogenase. Next question is, what is the biochemical basis of hemolysis? Here the biochemical basis is same. due to deficiency of enzyme glucose 6 phosphate dehydrogenase, HMP shunt pathway is going in a slow speed. Therefore, level of NADPH concentration is also low, and this NADPH is required to remove the hydrogen peroxide from the cell by enzyme glutathione reductase. And due to deficiency of NADPH, the hydrogen peroxide is not removed from the RBCs and they get hemolyzed. The next is how disease is transmitted. I have already told it is a sex-linked recessive disease. Write the reaction catalyzed by defective enzyme. Here you have to write down the first reaction, conversion of glucose 6 phosphate to 6 phosphogluconolactone by enzyme glucose 6 phosphate dehydrogenase, and the coenzyme is NADP, which is reduced to NADPH. Which precautions patient should take to avoid this complication in future? To avoid this complication in future, the patient has to tell to the doctor about his previous episodes of hemolysis, anemia, jaundice, so that the doctor should not prescribe him the drugs like primaquine to him, and he should not take the diet which induces oxidative stress in the body like fava beans, etc. This is about HMP shunt pathway and its related disorders and its significance. Thank you very much.